installment of the 2024 Amgen Biotech Experience Teacher Roundtable Series. My name is Reagan Davenport, and I work in the ABE program office. I'm the Community of Practice Liaison, and so it's a pleasure for me to host this roundtable today because I always appreciate the space to meet and see all of the wonderful folks that I hear about from the ABE program office, all of the wonderful things you're doing, and all of the wonderful things that your site is doing as well. So for those of you who are new to the series, these roundtables are designed for our incredible ABE teacher community from around the world. And it's a chance for all of us to get together to hear and learn from experts, from each other about topics of interest in science and biotechnology. So we hope you find this series valuable. And if you have any suggestions or ideas that you think would make a great roundtable topic, please email us at the ABE program office and let us know. We are in the process of planning future roundtables, and so we would love to have that input from you all who spend most of your time in those spaces. So this roundtable will be recorded and posted on our website, and you will also receive a transcript of the discussion that we have today if you're registered for this webinar. So we will have time built in at the end for audience questions, but we encourage you throughout the presentation tonight to put any questions that you have in the chat, and we'll be sure to watch for those and ask your questions at the end, okay? So I'm going to jump right in by introducing our amazing panelists. Melanie Rosenhart joins us from ABE, the Netherlands, and has done extensive work with educators in the fields of science education and human biology. She wants to integrate opportunities to build science skills at all levels, and she has been an incredibly valuable contributor to the ABE Netherlands program overall, and she assists her site by facilitating learning for teachers. We're also joined today by Silvia Vallecchio from ABE Italy, who currently teaches science at the G. Marconi Scientific High School in, is it Foligno? No?
Sorry?
I'm trying to say where your high school is at. I'm like, am I saying it? I'm probably not saying it correct. Is it Foligno? Foligno?
Liceo Scientifico Marconi Foligno, Foligno,
Okay.
Umbria, Italy.
Italy. Thank you, Sylvia. Thank you for helping me out there. Sylvia is passionate about working with students and providing them with opportunities for research in science education, and she also serves as the ambassador for the trainers at the ABE Italy site. So Melanie and Sylvia, thank you so much for joining us. We are pleased to have you both with us this evening because it is like 10 o'clock where they are joining us from for all of our US folks. So really appreciate them taking the time to come and have this discussion with us. So we're just going to begin by learning a little bit more about our panelists and their journeys as educators within science education. So Melanie, would you mind telling us a little bit more about your pathway into science education and your current role, including your work with ABE?
Yes, I will. So after I did my biology bachelor at the university in Amsterdam. And after that, I started with my master's in forensic science. And during my master's, I entered the science communication by doing as a student assistant, working at the university, but working actually at high schools and teaching them with science. And actually, during my job over there, I entered the DNA labs on the road. I'm a product leader over there now. But I started when I was a student and then became a coordinator over there. And so long. And then in 2016, we were approached by ABE, by the ABE program, because we already have a program going on in the Netherlands by itself. So we had a big network, and they approached us to collaborate with ABE. And yeah, we said yes about that. And then we entered the program in 2017. And in 2019, we started with the kids. And one of the kids is, do you taste PTC? And it's actually a precision medicine kit we now have for ABE. And we have now currently, official, we have 3, but we already have 4. Because we have last week, we have a new distribution site in the Netherlands, in the north of the Netherlands. So we have like 4 kits now. And all those locations, we do PDIs with the teachers, so teacher training programs, so they can land the kit and do the kit with the students at their school. The teachers are really enthusiastic because with this kit, they do, of course, the micropipetting, they do the gel electrophoresis, they do the DNA isolate extraction, the PCR, of course. They do everything by themselves with this kit. And the equipment is a little bit new. It's not like the older ones with the big PCR in the back of the classroom. No, they have we have mini PCR, and they are small PCR machines, and they have to install themselves, the students, so they are more, how you say that, in the lead of their own science, and they learn about their own genotype and about their phenotype, and that's really interesting for the students. They learn something from themselves. Hopefully that's the question.
It is, and I appreciate you. Thank you for sharing that, and congratulations on getting another distribution site up and running. I know that takes a lot of time and work, so congratulations on that.
Yes,
And
thank
it's
you.
great to hear that your students are what we would probably call over here like student-led learning. So they're leading their own learning, and they're going through the whole, inquiry process on their own and making those things relevant to their actual lives, which is where we
Yes.
know that they learn best anyway. So thank you for sharing that,
Yeah.
Melanie. Sylvia, how about you? Can you share your pathway into science education and your role in your work with ABE Italy?
Yes, good evening and good afternoon, everyone. And thanks for this opportunity to this professional condivision on AB precision medicine pathway. I'm a science teacher at Liceo Marconi, and I have a degree in biology, a PhD in medical histology and embryology, and a specialization in medical microbiology and virology. For 16 years, I dedicated myself to medical research in Italian university laboratories, focusing mainly in immunology. In 2008, I switched to teaching, and my passion for medical research and my desire to instill love for science in my students led me to collaborate with the ABE community. In this community, I found a great support for my teaching and professional development. In 2018, I started investigating the polymorphism of the TH2R38 gene in classroom using the Mini1 kit. And since 2021, I have been using the AB pathway on precision medicine, applying the ABC approach. I believe that the AB pathway on PM is very polyedric, so it allows us to address various complex concepts in a practical and not just theoretical way. This pathway allows us to eliminate misconception about genetic mutation or the relationship between genes and proteins. Thank you.
Thank you both so much for sharing that. And I don't know if you want to share this now or if you would rather share this when you each do a presentation of your work in precision medicine. But is there, or I guess why have you chosen to implement the precision medicine module with your students? I hear both of you talking about your backgrounds, and so it sounds like it is very relevant to those. But just for folks that may be listening that don't or aren't currently implementing the precision medicine module with their students, is there something or some things that drew you to that besides, both of your backgrounds, I guess? And how did your audience respond to that? I know, Sylvia, you teach, you work with students and teachers, and Melanie, I know you primarily work with the PDIs. So is there anything influencing you in that space, or were you both just like, we are brilliant and we have, which you are brilliant, have background in this area, and so like, yes, we're going all in?
Okay. We started with the kit before we lent out a kit in 2019. We already started before in 2018, of course, thinking of this. And if you all know, the ABE labs, the original ABE labs, the original series, contain were in the Netherlands not possible because of law and things like that. So we were thinking about to do something else, not to do with the GMO side way with transformation of bacteria. That was not possible in our classroom, so we had to think about something else. So we started a focus group with teachers. So we got input from different teachers, and we were thinking, what is important that those students learn during this practical? So, and PCR and gel electrophoresis and micropipetting was, of course, where those things were really important for them. So we were thinking what kind of practical was nice. And I think ABE didn't start at precision medicine yet. So I don't know how that happened, but so that's why we call it different. We call it like, do you taste BTC? So that's why we refer to a different name, but it's actually the practical is the same. And so we started with that actually, and also because the series is shorter. In the Netherlands, our teachers cannot do 5-hour or 5-day courses or something like that, because the original series take so much more time than this practical. So time-wise, we were thinking about these stuff. So this practical can be done in 5 hours with students. So, and that was a real deal breaker with our Dutch teachers. And and so we are now a couple of years later, and the kit is fully booked. So I think they like it. So, yeah.
Well, and that's wonderful to hear that. We know that teachers everywhere are always paying attention to the time and how long it's going to take and whether they have time and space to take it on. So I'm glad that they enjoyed it and that you say that it's staying fully booked. What about you, Sylvia?
I can share my presentation for
Go
just a
for
moment.
it. Yes, well, I know you both have a wealth of knowledge and insight into both of these areas, and so absolutely you can share your presentation.
Okay.
And remember, we're leaving Q&A time at the end, so if anyone has any questions, feel free to put them in the chat, and we will ask them at the end. So, Sylvia, you are
Can
up.
you see my screen? Can you see?
Yes, yes, we can.
Okay. Just a moment. Okay. 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 My ABE experience began in 2017 in Naples by attending the ABE course for teachers on RFP, RFP cloning. But in 2018, I started investigating the polymorphisms on the TAS2R38 gene in classroom using the Mini1 kit, a taste of genetic Minilab. So when Anna Pascucci in September 2022 asked me to do the ABE course for veteran educators on Introduction to Precision Medicine, I was very happy to do it. Excuse me, I go in presentation. Okay. Okay. My goals in this course, my goals were apply the IPSE approach to the ABE activity, choosing the level of inquiry based on the complexity of the experimental pathway. Help implement ABE activities in classroom and try to enrich ABE activities with a personal contribution.
Oh, Sylvia, I think you're on mute.
Okay.
Nope, there you are. There you are.
Do you? I restart my presentation.
Oh, we can hear you. I think the last I didn't hear anything you said toward the
In
end of
this
slide.
slide.
Yeah.
This slide, okay. In this course, or in this ABE course, my goal were applied IPSE approach to ABE activity, choosing the level of inquiry based on the complexity of the experimental pathway. After help implement ABE activities in classroom and try to enrich ABE activities with a personal contribution. But why the IPSE approach on precision medicine pathway? Because I think that the IPSE approach is important for the learning, to improve the learning on science in general. But in ABE pathway is essential because the laboratory ABE pathway are complex, not difficult, but they are complex. So students often focus on the technical practical aspect of the course, losing sight of the theoretical concepts and the overall framework of the course itself. While ABC approach with its well-structured phases simplify the process and consolidates the concept learned. In my presentation, for reasons of time, I will show only some of the numerous ABC activity that I did in this pathway on precision medicine. In the slide on the left, there are all the steps of the ABC pathway on precision medicine. And for first step, engage, I taught two types of engage added in two different moments of the activity. An engagement on genetics that aims to motivate and to recall the prerequisites, and then an engagement on precision medicine that aims to stimulate investigable questions. For the engagement on genetics, I taught to an educational video on Mendel law explained with lyrica operetta, and then to consolidate the theoretical concept, I added this sample group activity on the transmission of the light eye colors. In particular, this activity asked starting from the parent's genotype, calculate the probability of having a child with a light eye. After this recalling of the prerequisite, it is important to explain what SNPs are and to facilitate the learning of this concept. I inserted a different activity, one activity for each one type of SNPs. These are all the activity. In the first activity is on silent SNPs. In this activity, students understand that a changing in a single base of DNA not always lead to a change in a protein. In the second activity on silent SNPs is an activity on SNPs that cause harmful variation, such as detached or attached earlobe. And in particular, in this activity, the students, with reference to the phenotypic variation examined before, we evaluated the dominant recessive phenotypic characters present in the class group and calculated the percentage of expression. In the last activity, I did an activity on SNPs that cause disease, such as sickle cell anemia, and the students, through the electrophoretic profile, have to identify the healthy carrier and the sick subject. After this first engage on genetics, I introduced an engage on precision medicine. And in this engagement, my focus were to associate the different perception of the bitter taste in response to PTC with a complex traits such as a different response to drugs, predisposition to disease. For this engagement, I taught three steps. In the first steps, students have to taste the PTC paper, and they have to classify their phenotype in taster, medium taster, and no taster. In the second phase, I taught to a brainstorming on the different reasons responsible to the different perception of the bitter taste. The students have to classify their phenotypes in associate their phenotypes to a kind of SNPs, no sense mutation or sense mutation, and then they must to guess your genotype. After to connect the experimental pathway on PTC to precision medicine concept, I had students to read a different scientific paper on pharmacology of bitter taste receptor and the role of bitter taste receptor in airway immune defense. At this point, the students have all information to develop or answer to an investigative question. This is an example of investigative question. How can it be demonstrated that the different perception of bitter taste depends on a mutation on bitter taste TS2R38 receptor? The students have to formulate their hypothesis. Also, the experimental activity is carried out with IPSE approach. And to facilitate this approach, I divided the the entire pathways in four phases: DNA extraction, the amplification of the TS2R38 gene, the digestion with the restriction enzyme, and electrophoretic run of the digestate. To improve the learning of the theoretical concept from the single phases, I have introduced investigative paper activity, one activity for each experimental step. This is an example of investigative paper activity on prediction. In the slide on the left, there is the hypothesis of the students. In particular, the students, the activity asked, based on the previously hypothesized genotype and collected data, represent the electrophoretic profile you expected to obtain. On the right, compare the data obtained after the electrophoretic run with your prediction, identify your genotype, and trace the size of each band. To complete the process of learning, I added at the end of the experimental phase the simulation on Lab Exchange platform to improve the learning of the entire process. And after I introduce a construction of a paper model of the TS2R38 receptor. In some cases, the data obtained can be contradictory, testifying that the perception of bitter taste as the sensitivity to a drug is a multifactorial variation due to both genetic and anatomical environment factors. So, new research question was born. For example, what other factor may be responsible for the different perception of the bitter taste? In the previous brainstorming, students referred to the taste buds on the tongue. So we have detected the number and the shape of the fungiform papillae on the tongue, responsible for the different perception of the bitter taste. On the left of the slide, there is the methodology, and on the bottom, there are the different ranges for the classification. At the end, we have cross the data and draw conclusion. We have evaluated how much the results agreed with the frequency of the Caucasian population reported in the literature, that is around 13%. These are my students during the activity in laboratory, and that's all. Thank you, ABE, for this contribute to my teaching.
Thank you so much for sharing. And there's so much to unpack there, but I love how you are supporting your students through the process and how you're making it relevant to them. And I liked your slide where you were like, oh, now we have a new research question, sort of showing them how it evolves. And although we're looking for this specific answer, right, like we're going to go about it in a number of ways. So I appreciate that. I always love seeing pictures of the students as a former educator myself. So thank you for sharing that. That was amazing. What about you, Melanie? I know that you work a lot with educators. So would you mind sharing with us now your experience with the precision medicine?
First, I was unmuted,
Oh,
of course.
it's
Yeah.
fine. Zoom light.
Yes. Of course, we have a similar power presentation during our PDI like Sylvia, because we want like that the teachers experience the practical themselves as well. So they go through the whole practical by themselves because some teachers didn't micropipette years before or something like that, or never did something like this, the PTC tasting. So for them, it's new. Also, the equipment is new. We are using mini PCR, but I'm also familiar with mini one, so kind of similar, both from the US. Small, I call it doll science because it's all small. I have some pictures in slideshow as well about our kit and also how our teachers are working inside the lab. The presentation you already saw, we have something similar. We talk about the SNPs, of course, how they involve all the science behind it. We also have a small video about tasting because it is some people are hydrozygotes with tasting, but some people are really, if they taste a PTC paper, they're really crying or because it's so bad. And some people are, oh, nothing happened, but they're still homozygous staters. So how does that happen? And so we have a clip, it's in Dutch, so will not show it, but they talk about tasting and all around the world. And yeah, if you're familiar with kind of tasting, you maybe grow on it, so you're used to and stuff like that. So it's good to have multiple opinions, but I will share now. So I will show you some pictures and we'll go on the slideshow. So this is how they work in our, this is in Amsterdam. So you see teachers working. And actually this teacher over here was my old biology teacher. So that's quite funny that she is in my PDI. So they work always together and they have, you can see, do you see my mouse? Yeah, okay. So this is the mini PCR and they are doing something on the eyes. I think they are doing the PCR setting in. They all work with mobile phones to put on the PCR. Nowadays in the Netherlands, the high school kids are not really allowed anymore to have their mobile phones in the class. So we have to shift with that. This is just our PCR. Yeah. And then this is me presenting and our different distribution site. This is in Delft. You can see the blue gel from a mini PCR as well with the little doka on site. And this is in Nijmegen. And they are practicing with the practice gel before every step before they actually do something. We let our teachers practice before they actually insert something into the gel or in tubes, because what I already said is some teachers maybe micropipette years before, so for them it's also new, just like students actually. So we just let them learn. And you can, if you see in the back, you can already see our kit in boxes, the blue kits. So they will do everything and we show them some steps and we present a little bit more on the board. So it's like do something and we do a lecture kind of thing. And also give them during the PDI, of course, we also give them heads ups. So this is maybe if you have 20 students, you can maybe do something like that. If you have 30 students, if you want to auto quote, you can do it like this. So we give them handouts what to do in their own class situation. And this is I will show our kit. This is so if teachers come collect our kit, this is what it looks like. It's containing in 3 boxes, and they are like stroller suitcases, so you can roll them. You can see here the 4 PCRs and 4 gel electrophoresis, and the casting, the gel casting, and then of course the tips, the racks, the pipettes, and the centrifuge, the tasting paper. We use the toothpicks to collect saliva sample, buccal swab, and this is a mini cooler where all the reactions will go in. So it's up to minus 20 degrees Celsius. I don't know how much it is in Fahrenheit, but it's to keep it frozen. And we get the mini PCR kit, and that's with the agarose and the tips and something like that. So the teachers, they book a time slot, they come to us, they pick it up at one of our distribution sites, and they go to their school and put their reagents immediately in their freezer. They can loan the kit for like 3 weeks or maybe 4, and they can get 1 kit or multiple kits, depends on how many students they have, of course. And they will return the kit again, and then we all setting everything ready for the next teacher who will loan. And in the beginning or the end of the year, we will give a PDI, so they can attend to one of the PDIs, and we try to do the PDIs all around the country so they don't have to travel much farther. So we are in the Netherlands, but we also have some Belgian people. So from Belgium, they also speak Dutch. They will lend our kit as well. So it goes also abroad. It's not a small country, but they go abroad as well. So this is our kit. Yeah, I have some pictures more to zoom in. This is how it looks. Yeah, I don't know, the mini PCR, you just put the PCR tubes inside. It's not that spectacular. And this is how it looks like. And it's also very handy with the foam. The teachers can see how they handle, how to do to put stuff back. And it's just easy for everyone. And this is how my slideshow a little bit. Yeah, and after they return the kit, the teachers get an evaluation. So if maybe they have tips for us or not. Yeah. So that's it. Well,
Thank you. Thank you for sharing. So there is a lot that goes in to this precision medicine lab. And I'm just wondering if either of you or both of you have, I know Melanie, you just spoke about the evaluation that you give to teachers, and you say, okay, do you have any tips for, how we can maybe organize this better or make it more, easier for you to access? Do you find that you get feedback from the teachers, like their responses? Are they asking for the same thing? When you say tips, do you find there's a sort of like a theme or a big idea that they're sharing that you all have integrated into getting the kits distributed?
Now, actually, mostly we wrote our manuals and how to do the stepwise to the program, and sometimes they said maybe you can write it a little bit easier. But we also notified, we use kits from mini PCR, and they change sometimes the insides of the reagents, so it looks differently. And then I thought, okay, they changed it like in 1 year, they changed it 3 times or something. So I was like, okay, I need to rewrite again, and this is not handy for us. And then, if we order something from them, what does it look like? What is it, something new? Do they have to add the gel green, or is it already in the tabs? Because now they already integrated in the tabs, but before they had it not integrated. So you had a different work-wise to prepare a gel, and that's very important if the manual is actual is correct. So that was in the beginning, it was a thing, but for now, some teachers are, I don't know, during the PDI, I don't know if you saw the casting tray of the mini PCR, but they have the slots to make slots in the gel. It's underneath the casting tray. And I was called by teachers, where I looked everywhere in the kit, where are those slots? I was like, yeah, they're underneath the casting tray, sorry. So you just need to swap. So we had that a couple of times, but that's the easy one. So that's, but not for, no, maybe Sylvia, maybe you have.
I was gonna say, Sylvia, do you have any tips or something that your students and teachers are asking for when you start to use the precision medicine lab?
My students have appreciated the digestion with the restriction enzyme, this phase, and the related electrophoretic run, because they were able to visualize the results of the entire experimental phase, and verify their hypothesis. This is very important. Also, the activity on the taste buds on the tongue is very interesting, but also very, very fun. They are at the end had the all blue tongue, and while the investigative ABC activity were exciting but difficult, because these activity are different from usual teaching methods. In all experimental phases, I have not problems, because also the kit that I have used is very simple to use, and the activity of the teacher is focus on the gestion of the class, of the students. And to simplify the process, I divided the complex of the pathway in the different phases, because is more simple, is more simple. And introducing the ABC paper activity improve the learning of the theoretical concept, the abstract concept, and so carry out each phases with the IPSE approach, simplify the process, the experimental process.
Can I
That
ask
is,
a practical
I'm sorry. Yes.
So in the Netherlands, are you doing the regular ABE labs with the restriction digest and the ligation? I know they are in Italy. So practical thinking, I want to know when your teachers are incorporating this, because this seems like an awful lot of material to cover. Our teachers get the kits for 3 weeks to do the main foundations of biotech labs. And so are they doing this as a standalone later in the semester? Are they tacking this on after the foundations labs? How are you doing this? Oh, Melanie, you're muted.
Oh,
It's
sorry.
okay. It's okay.
Okay, Karen, a lot of questions in one question. So what's your first question?
Are your teachers doing the foundation labs as well as this?
Not the teachers, the student. I have students as a master class. They will do the normal ABE labs.
Okay, so are
With
these
you mean
are
with the
these
transformation? You mean like that?
right. So I'm assuming, let's say teachers don't have their own equipment. So they're going to have to borrow equipment to run the foundation labs as well as for this EPM lab section. So that's why I was asking how long it was taking your teachers to get through this, because if I'm giving out my kits for 3 weeks, I'm not sure they have time to tack this on.
No, yeah. So we only use the kit for the precision medicine.
Okay.
And that depends on how many classes they will do. So we have a teacher who books, but they can do multiple classes, and those classes have different times of schooling, of hours. So they have to, arrange that in those 3 weeks. Because the time slot of 3 weeks is also including to collect and to bring back the kit. I don't know if
Okay, and Sylvia, is that the same with you, that your teachers, are they also doing the foundations labs and adding this on at the end? Or doing it at a different time?
I don't understand, excuse me.
So your teachers, they do the foundations of biotech labs?
Yes, also another teacher are present in my school that have that collaborated with ABE pathway.
Okay. Now, do they do the precision medicine after they're finished with those other labs?
Yes, also
Okay.
in precision medicine. Yes.
Okay.
okay,
Three
Karen's
teacher,
yeah.
also 3 teacher.
Okay. So Melanie told me in the chat that it took her teachers about 5 hours to cover the lab materials and the some of the concepts for EPM, but yours looked a lot more involved. How long did it take your teacher, Sylvia, to cover this entire module? Okay. Yeah.
So for I will maybe for our teachers, I will teach them what they could teach, and of course during when they teach the precision medicine in their classes, I don't know how long it will take. Maybe they do multiple hours more to get the concept right about the genotypes and the phenotype and the SNPs and stuff like that. I don't know how much time they will take, but for all the lab work and some basic concepts, it takes like 4 or 5 hours. And maybe they along it with like a presentation, what Sylvia already does a little bit more. We have also the presentation, but if they want to teach something sideways, they can always, it can take longer. And to the other question, we don't do the foundation labs in our school because we're not allowed, but we give the opportunity to students to come to the university to do them.
Okay.
So it's a whole different, program for us to run.
Okay. All right.
So maybe now can Sylvia say how long their teachers will teach about EPM.
And I understand. I do the experimental phase in 11, 20 hours in the laboratory.
Okay.
In 12, excuse me, 12 hours.
Thank you.
And I actually was wondering, Sylvia, do you use a kit from Mini One? For
Yes.
precision? Okay.
The kit of Mini One, the Taste of Genetic Media Labs is the kit more that have the digestion, the DNA extraction, and amplification is complex,
Yeah.
more complex. Then there is a kit no more complex, only the electrophoretic run of the sample just treated.
Okay. And how old were your students on the picture? Are there?
Yes, in my presentation, there were my students, and the students are 24 each time, and of the first class, the 5 class of the high school, the last year of the school.
The age is then 17 till 18 or something?
Yes, 18, 17, 18.
Yeah, okay. Yeah. So we use the kit, our teachers use the kit for, I think, also similar 16 till 18 years old. And we also say that if they have the kit in their school, notify to other teachers from different classes, like ages from 11 till 14, you can do the pipetting classes for them as well, to learn about pipetting. If you already have all those micropipettes, you can just do the micropipetting courses with them. We have them in the slides, in the manuals. You can do them already with them, with the younger kids. But then the teachers need to talk to each other, and that is there.
Also, I use always the first 2 laboratory of the ABE pathway on RFP cloning, because it's important to teach the use of the micropipette and the electrophoretic run.
Yeah.
So I use always the first 2 laboratory.
Yeah, and we also send our teachers and students and their students to lab exchange as well, if they can already learn about all those basic concepts as well.
I love this conversation. We have so many wonderful. So first of all, thank you, Karen, for asking that question, because it has opened up a really good discussion about what portions of the lab are being covered, how they're being covered, the time frame for them being covered. I know that I was just at a meeting for ABE with a group of teachers, US teachers, and they were talking about, you know, sort of some of the challenges that they have around getting them set up and like to the distribution sites and getting them, you know, to teachers and getting them back from teachers, and some of the things they can do and some of the things they can't do, like you alluded to, Melanie. And so this is helpful because I'm hearing some of the things that you're saying about, you know, this is how we do it, or you saying that like students can go and do the portion that they can't do in school, they can go to the university and do it. And so it's very helpful. And I think teachers in the ABE community, they'll appreciate knowing this, because that's sort of one of the things that I feel like I hear a lot about the precision medicine lab, that there's all these workarounds and they're not really sure how to work around them. And so thank you again, Karen, for asking, and thank you, Melanie and Sylvia. And I see Anna in the chat offering up what it looks like with your audience, with your students and with your teachers, and how it looks at PDIs and different things like that. So I appreciate that so much. Does anyone else have any questions for either Sylvia or Melanie that they would like to have answered?
So I'm going to ask just one practical
Okay,
last question. So we just got through a giant pandemic, and do your students have any limitations about putting toothpicks in their mouth? Are they still, obviously they're allowed to do that in your country, and dispose of the waste in the trash there? Yes.
So, sorry, what?
So everyone here is really paranoid about, no, we can't have them put PTC paper on
Yeah.
their tongue, and we can't have them use a toothpick. And I'm like, we did it just before. There's no COVID. Why can't we do it now? But so obviously you guys are able to do that. The kids are tasting the PTC themselves, and they are scraping and extracting their own DNA.
Yeah, but if they're with their finger in the mouth, they will smear saliva everywhere, anywhere at all too. So I don't know if you want to do a saliva kit somewhere. I will happy do it the forensic kit I have with saliva. No, but no. So, but in the Netherlands, actually, we never heard about that in school. Yeah. Bacteria transformation, they say no in the Netherlands.
Yeah, we can do that, but you know, we can't have students put a piece of because I
No.
think that the PTC, them tasting it themselves and seeing it on the gel, that's the most aha moment for the students. Yeah, that's what makes
I
it
know that
personal.
Germany, it's not allowed to do the PTC tasting in their mouth because they put chemicals in their mouth. Yeah, I think teachers believe us that we're good people and we don't lend out stuff that it's not good for their students.
That's
I don't know.
harmful for
I mean,
children,
a
right?
student could always decline if they wanted to, but yeah.
Yeah, instead if they don't want to do it, they will not book us. I think that's the whole thing. They don't book our kit. And we enclose with the PTC paper, we will enclose what it is, so they can read what it is. So
okay.
And with the toothpick, they scrape their mouth. We always say don't do it too hard, that we don't want to scrape blood or something, or just do it gently, but scrape enough, because if you don't do it right, then you don't have DNA. So that's you want DNA. But I also saw something in the chat about the bioinformatics. I know that in the model you can also sequence your own DNA. We don't do that. I think that's time consuming and price wise as well.
Right. Yeah, I think we dropped that, but they just provide sequences for the 3 different phenotypes, and then they look at that.
That's a good thing. And we also compare them with other animals as well, because other animals have them as well, those SNP. So so that's also interesting why you have this PTC tasting and why you don't. And
it is always very interesting.
Yeah.
Would you be willing to share that information on the other animals? I mean, do you look at things like herbivores versus carnivores? That'd be kind of interesting.
I don't know. I know that there's some primates on. I saw the gorilla, the orangutan. I don't know. I don't know if there are carnivores on it as well. I don't think so. It's because of fruits. It's
right. I know. That's why I was wondering if they have it at all. That's interesting. So if you do have a link to that, maybe you could email it.
Yeah, I have it in my presentation. I can share your.
That would be
because
great.
it's from a bioinformatics thing. It's I think it's from BLAST.
Okay.
They blast it, so if you can blast the SNP part, you can see what kind of other animals have this as well. So
that's interesting. Thank you. Look at all of the thought, what all the thought partnering is producing. This is amazing. Debbie had a quick question because I know we're about 2 minutes over time, so I don't know if folks can stay on for another 2 minutes. But Debbie had a question for you, Sylvia, and it says, when Sylvia has her kids compare numbers of taste buds to research data, do you have your students do any statistical testing? And if so, what kind? You're on mute, Sylvia, sorry.
Okay.
Yay, now we can hear you.
I use in general when I do this taste on this test on taste buds, I use a disc of paper to obtain the same surface where I count the number of the buds or the papille fungiformi, and I saw that the number is the same for each type of phenotype. In general, the no taster have from 0 to 10, 11, 12 papille. From 11 to 30 are taster, medium taster, and up to 30 papille are super taster or taster. I do this test since 2021. And the number is not always the same, but is similar, very similar. And the frequencies that I have found is always the same of the frequency obtained in literature from Caucasian population. It's very interesting to compare the electrophoretic run, the data of the electrophoretic run and the number of the papille. But is not only these reasons for a different perception of the bitter taste, because there are other factors, also protein in the mouth, in the saliva, that can inhibit the sensitivity to a bitter taste. There are many factors that influence the perception of the bitter taste. Not only genetics, not only the papille fungiformi, but environment factors in the mouth.
Thank you so much. And I feel like that's such a good example for running the lab across many classes, or every year, keeping your data and just building a data set that your kids can really analyze. But thank you so much. That's such a great idea. Thank you.
I do on every years with different classes, and I do this test since 2021. So is are 3, 4, also 5 years that I do this test.
Great. Thank you so much. Great question, Debbie. Like I said, there's so many things that have come about from this discussion. So as we come to the end of our time today, we're a little bit over, but this was such a great discussion. It's worth it. I always want to say a final thank you to our panelists, Melanie and Sylvia. Thank you for joining us in the wee hours of the night over there. We hope this roundtable was informative and gave you some ideas for how you can implement the precision medicine lab in your own classrooms. You're welcome to visit us at the ABE website anytime. Sarah, I don't know if you can throw that email in the chat again for me. And again, please stay tuned for more information about future roundtable discussions and how you can participate. This is making me think maybe we need a part 2. So I am going to have some conversation with the program office and see what they think about that. But as we know, the ABE teacher community, you all make the program thrive and grow. And so we want to thank everyone for being here, everything you're doing for educators and students worldwide. And as always, we like to thank the Amgen Foundation for supporting the Amgen Biotech experience. I hope that you all have a wonderful rest of your week. And thank you for joining this ABE Roundtable. Bye, everyone.
Thank you so much.
Thank you so
so
much.
much. Happy Easter.
Happy Easter.
Happy Easter.
Take care.
And could you share your presentation as well, Sylvia? Maybe I would like to add the taste buds testing as well. Yeah, it sounds weird.
that.
Taste, yeah.
Thank you all so much.